bacterial transcription, and its regulation. In this chapter, we aim to explore the essential mechanisms of bacterial transcription. By the end of this lesson, you should be able to explain the stages of transcription in bacteria, describe the structure and function of bacterial RNA polymerase, and analyze how transcription is regulated, particularly through feedback loops, regulatory sequences, and operons. Bacterial RNA polymerase is a powerful enzyme capable of synthesizing RNA using a DNA template and 4 nucleotide triphosphates. Remarkably, unlike DNA polymerases, it does not require a primer to begin synthesis. The enzyme catalyzes the addition of nucleoside monophosphates to the growing RNA chain in the 5' to 3' direction. This process occurs in 3 defined stages: initiation, elongation, and termination. To understand transcription, it's important to clarify some key terms. The template strand is the DNA strand that serves as the pattern for RNA synthesis. The coding strand, also known as the sense strand, is complementary to the template strand and matches the RNA sequence, except for uracil replacing thymine. The transcription start site is labeled +1. Sequences downstream of this site are given positive numbers, while sequences upstream are assigned negative numbers. This figure illustrates how nucleotide positions are numbered on the coding strand. Understanding this numbering system helps to pinpoint promoter elements, transcription start sites, and regulatory sequences. Bacterial RNA polymerase is a large multi-subunit complex consisting of 6 subunits: 2 alpha, 1 beta, 1 beta prime, 1 omega, and 1 sigma. The core enzyme, composed of the 2 alpha subunits, beta and beta prime, can synthesize RNA using single-stranded or nicked double-stranded DNA, but cannot function with intact double-stranded DNA. The sigma factor is essential for promoter recognition, but not for catalysis. Together, these subunits allow the polymerase to recognize and transcribe thousands of genes. Here, we see the crystal structure of the T7 RNA polymerase bound to DNA. The polymerase is divided into 3 colored subdomains: red for the palm, green for the thumb, and orange for the fingers. These regions collectively form the active site responsible for RNA synthesis. To begin transcription, the RNA polymerase must recognize a promoter sequence on intact double-stranded DNA. This requires the holoenzyme, which includes the sigma factor. The sigma factor plays a crucial role in identifying the promoter, enabling transcription to start at the correct location. In E. coli, the primary sigma factor is sigma 70, which regulates essential genes. Sigma 70 promoters typically contain 2 conserved sequences: the -10 box, also called the Pribno box, with a consensus sequence of tatat, and the -35 box, with the sequence tetgaka. The ideal spacing between these 2 elements is about 17 base pairs. Promoters that closely match these consensus sequences are usually stronger and more efficient at initiating transcription. When the holoenzyme binds to DNA at 0 degrees Celsius, the DNA remains double-stranded. This is called a closed promoter complex. However, at physiological temperature, around 37 degrees, the DNA unwinds from position -12 to +2, forming an open promoter complex. The at-rich -10 box facilitates the melting of the DNA strands. The RNA polymerase resembles a crab claw in shape. The 2 alpha subunits dimerize and associate with the beta and beta prime subunits to form the active site. Within the beta prime subunit, the bridge helix and trigger loop are essential for adding new nucleotides during RNA synthesis. Rifampicin is an antibiotic that targets bacterial RNA polymerase. It inhibits the initiation phase of transcription, making it a potent tool for treating bacterial infections like tuberculosis. This highlights how fundamental understanding of transcription can inform drug design. During abortive initiation, the polymerase frequently releases short RNA fragments, typically 8 to 10 nucleotides in length. To transition into elongation, RNA polymerase must overcome promoter binding through a process called promoter escape. The scrunching model explains this phenomenon. The enzyme pulls in and compacts DNA, storing energy that is later used to break free from the promoter. Once elongation begins, the polymerase forms a transcription elongation complex. This complex includes the core enzyme, the DNA template, and the growing RNA strand. The structure ensures processive RNA synthesis along the DNA strand. This schematic shows the internal organization of the elongation complex. The DNA is threaded through the polymerase, and the RNA strand emerges from a separate channel. This arrangement supports stable transcription and protects the nucleic acid intermediates. During elongation, each nucleotide triphosphate must enter the active site of RNA polymerase. This is the rate-limiting step, since only 1 out of 4 nucleotides will be the correct match. Once incorporated, a phosphodiester bond is formed, and the polymerase moves 1 nucleotide downstream. Although it synthesizes RNA at roughly 30 nucleotides per second, it frequently pauses, allowing regulatory proteins to bind and coordinate transcription with translation. RNA polymerase has proofreading ability. When an incorrect nucleotide is incorporated, the enzyme pauses and may backtrack. Mismatched nucleotides can be removed either by the polymerase itself or with the help of elongation factors such as GRE and GREB, which cleave short stretches of RNA to restore accurate synthesis. Bacterial transcription terminates through 2 main pathways: intrinsic termination and rho-dependent termination. Intrinsic termination depends on specific RNA sequences. These include a GC-rich inverted repeat that forms a stem loop structure, followed by a stretch of uracils. These motifs cause RNA polymerase to pause and dissociate from the DNA, effectively ending transcription. In contrast, rho-dependent termination requires a rho factor, a helicase that moves in the 5' to 3' direction along the RNA. It binds to a RUT site, which is a C-rich region of the RNA. Once bound, rho uses energy from ATP to catch up with the RNA polymerase, destabilize the transcription complex, and terminate transcription. In bacteria, transcription and translation occur in the same cellular compartment. This means that translation can begin at the 5' end of the mRNA even before the 3' end is finished transcribing. Bacterial mRNAs are often polycistronic, meaning a single transcript can code for multiple proteins. These transcripts have short lifespans and contain untranslated regions at both ends. Each coding sequence, or cistron, includes start and stop codons for translation. Bacteria regulate transcription through 2 major mechanisms. Negative regulation involves a repressor protein that turns off transcription. In positive regulation, an activator protein enhances transcription. Both mechanisms are critical for controlling gene expression in response to environmental changes. In E. coli, the genes LACH, LACI, and LACA encode enzymes for lactose metabolism. These include beta-galactosidase, which breaks lactose into glucose and galactose, lactose paramase, which imports lactose, and a transacetylase, whose function is less clear. These enzymes are not produced unless lactose is present in the environment. In the absence of lactose, The lac enzymes are present at extremely low levels, just 1 or 2 molecules per cell. When lactose is added, expression of all 3 genes increases dramatically to over 100,000 molecules per cell. These are known as inducible enzymes because they are turned on by the presence of an inducer, lactose. The lac operon explains how this gene regulation works. The LACH, LACI, and LACA genes are transcribed as a single polycistronic mRNA. Upstream, a repressor protein encoded by the LACI gene binds to a region called the operator to block transcription. When lactose is present, it acts as an inducer, binding to the repressor and releasing it from the operator, allowing transcription to proceed. The figure on this slide shows the lac operon model. It visually represents the interaction between the promoter, operator, and the lac genes. This operon system is a classic example of negative regulation through de-repression. 2 major problems were discovered in this system. First, lactose must enter the cell to act as an inducer, but lactose transport requires permease, which is itself only produced after induction. Second, lactose does not directly bind the repressor. These problems are solved by basal expression of the operon, which keeps small amounts of beta-galactosidase and permease in the cell even in the absence of lactose. These small amounts convert lactose to allolactose, the true inducer of the lac operon.
The expression of genes can be transcriptionally regulated. Such regulation is a critical feature of both eukaryotic and prokaryotic organisms. One of the classical systems used to investigate transcriptional regulation was originally investigated by Jacob, Monod, and Pardee. Their studies involved the transcriptional regulation of the genes used to ferment lactose in the bacterium Escherichia coli. 3 genes clustered together on the chromosome are required for the bacterium to utilize lactose. These were determined to be the LAC-Z gene that codes for the enzyme beta-galactosidase, the LAC-Y gene that codes for a lactose permease, and the LAC-A gene that codes for the enzyme galactoside transacetylase. The LAC-P region is the promoter that is needed to transcribe the LAC-Z, LAC-Y, and LAC-A genes as a single polycistronic mRNA. The LAC-O region is an operator site that is involved in the transcriptional regulation of the lac operon. The unit consisting of the lac promoter, lac operator, and the LAC-Z, LAC-Y, and LAC-A genes is called the lac operon. Located near but not in tandem with the lac region is the LAC-I regulatory gene that codes for an mRNA that is translated to produce a protein referred to as the lac repressor. In the absence of lactose inside the cell, the lac repressor protein is active. In its active state, the lac repressor recognizes and binds to the lac operator site. When the lac repressor is bound to the lac operator site, RNA polymerase is prohibited from recognizing the lac promoter, and the LAC-Z, LAC-Y, and LAC-A genes are not transcribed. When lactose enters the cell, a small amount of it is converted to allolactose via beta-galactosidase. Allolactose binds to the lac repressor. This causes a conformational change in the lac repressor protein that prevents it from binding to the lac operator site. Inactive lac repressor is unable to bind to the lac operator site. Without an active lac repressor bound to the lac operator site, RNA polymerase is able to bind to the promoter, and a polycistronic messenger RNA is transcribed that includes the LAC-Z, LAC-Y, and LAC-A genes. This polycistronic messenger RNA is then translated to produce the beta-galactosidase, lactose permease, and galactoside transacetylase proteins. Therefore, the induction of the lac operon enables the bacterium to efficiently transport lactose into the cell and metabolize it.
Researchers use IPTG, a synthetic analog of allolactose, to induce the operon in the lab. Unlike lactose, IPTG is not metabolized, so it remains active as an inducer throughout the experiment. This allows consistent, and sustained induction. The lac operon has three operator sequences. LACO1, located at position +11, is the primary operator. LACO2 is downstream at +412, and LACO3 is upstream at -82. These operator sites allow the repressor to loop the DNA, increasing repression efficiency. The lac repressor is a tetramer made of two dimers. Each dimer can bind one operator. The repressor contains several functional domains: a helix-turn-helix motif for DNA binding, a hinge region, a ligand-binding domain that interacts with allolactose or IPTG, and a tetramerization helix that helps the subunits assemble. E. coli prefers glucose over lactose. When both sugars are present, the lac operon remains off until all glucose is consumed. This phenomenon is called catabolite repression. Glucose presence lowers levels of cAMP, a signaling molecule that is essential for activating the lac operon. Glucose reduces cAMP levels through multiple mechanisms. Primarily, it inhibits the activity of adenylate cyclase, the enzyme that synthesizes cAMP from ATP. This mechanism ensures that E. coli does not waste energy expressing the lac operon when glucose, a more efficient energy source, is available. The catabolite regulator protein, or CRP, forms a complex with 3', 5' cyclic AMP, commonly known as cAMP. Together, they act as a positive regulator of the lac operon. For this to happen, the enzyme adenylate cyclase must convert ATP to cAMP. Once formed, the cAMP-CRP complex binds to a specific site on DNA and activates transcription. This system is crucial for increasing transcription of the lac operon when glucose is no longer available. This figure shows the cAMP receptor protein, or CRP, bound to DNA. Each monomer of the CRP dimer binds two molecules of cAMP. The binding of cAMP induces a conformational change in CRP, allowing it to interact with the major groove of the DNA. This interaction stabilizes the binding of RNA polymerase to the promoter and facilitates efficient transcription initiation. The cAMP-CRP complex binds to an activator site upstream of the lac promoter. In the absence of this complex, the lac promoter is quite weak because its -10 sequence poorly matches the consensus. The cAMP-CRP complex improves transcription by making direct contact with the C-terminal domain of the RNA polymerase alpha subunit. This interaction strengthens RNA polymerase binding and promotes the initiation of transcription. This slide summarizes catabolite repression. When both glucose and lactose are present, E. coli represses the lac operon. This prioritizes glucose usage, since glucose is a more efficient energy source. Once glucose is depleted, cAMP levels rise, allowing the cAMP-CRP complex to activate the lac operon. This dual-layer regulation ensures efficient resource management by the bacterial cell.
In prokaryotes, gene expression is often grouped into units of related genes and their control elements. These units are known as operons. One of the most well-understood operons is the lac operon, the set of genes controlling lactose metabolism in E. coli bacteria. Lactose is a disaccharide consisting of galactose and glucose. In order for a bacterial cell to make use of lactose as an energy source, it must cleave the disaccharide in two, a job performed by the enzyme beta-galactosidase. Production of beta-galactosidase when the cell does not need to use lactose would be a waste of energy and should be avoided. Therefore, the lac operon is only turned on under specific conditions. Beta-galactosidase and two associated genes, permease and transacetylase, are only transcribed when lactose is present. Because the lac operon is off by default and turned on only in particular circumstances, the lac operon is said to be inducible. The mechanism of this inducible activity merits further consideration. Within the lac operon, a regulatory gene codes for a repressor molecule. In the absence of lactose, this repressor molecule binds to the DNA just upstream of the genes coding for beta-galactosidase, permease, and transacetylase. These genes are known as the structural genes. The site of repressor binding is known as the operator. As long as the repressor is bound to the operator, RNA polymerase cannot proceed through the structural genes, and therefore transcription of beta-galactosidase, permease, and transacetylase is stopped. In the presence of lactose, allolactose, an isomer of lactose, binds to the repressor molecule and causes it to change shape. In its new conformation, the repressor molecule cannot bind the operator. With the operator free, RNA polymerase is free to bind the promoter and proceed with transcription of the structural genes. In the end, beta-galactosidase, permease, and transacetylase are produced and utilized to break down lactose for the cell to use. However, repressor-mediated transcription control of the lac operon only tells half of the story. Even if lactose is present, beta-galactosidase, permease, and transacetylase will not be produced if another, more desirable energy source is available. Because glucose is preferable to lactose, it would be a waste of energy to produce the tools for dealing with lactose, only then to use glucose instead. Lac operon expression also relies on positive regulation. In order for RNA polymerase to effectively bind and transcribe the structural genes, an active cAP protein complex is required. However, the cAP protein is only active when bound by cyclic AMP. When glucose is available, cyclic AMP levels remain low and CAP therefore remains inactive. Under these circumstances, even though lactose is present, little transcription of beta-galactosidase, permease, and transacetylase occurs. Only when levels of glucose are low is cyclic AMP available to bind and activate cAP, which in turn facilitates transcription through enhanced RNA polymerase binding. Therefore, cAP is active and the lac operon transcribed efficiently only when lactose is present and more favorable energy sources are absent. This mechanism for gene expression control ensures that cells do not waste energy making proteins that will not be used. The Operon system enables a prokaryotic cell to tailor its gene expression to the specific needs as dictated by its immediate environment.
The tryptophan operon, or TRP operon, is another classic example of gene regulation in E. coli. It is regulated at multiple levels, including transcription initiation, elongation, and termination. The operon contains a promoter, operator, leader sequence, attenuator, and 5 structural genes: TRP, TRPD, TRPC, TRPB, and TRPA. Together, these genes encode enzymes for tryptophan biosynthesis. Since the operon is anabolic, not catabolic, it does not have a cAMP-CRP site. Instead, it uses both coarse repression through the operator and fine-tune control via attenuation. This slide illustrates the enzymes encoded by the TRP operon and the biochemical reactions they catalyze. Each gene in the operon contributes to the multi-step biosynthetic pathway that produces tryptophan from precursor molecules. The pathway begins with PRPP, or phosphoribosylpyrophosphate, and proceeds through intermediates catalyzed by the enzymes TRPE, TRPD, TRPC, TRPB, and TRPA. This coordinated gene expression ensures that the cell can synthesize tryptophan only when needed. The TRP operon is turned off when tryptophan is abundant. Tryptophan acts as a co-repressor by binding to the TRPRA protein, forming a functional repressor complex. This repressor binds to the operator region, blocking RNA polymerase from transcribing the structural genes. In the absence of tryptophan, the aporepressor remains inactive and transcription proceeds. This is a classic example of negative feedback inhibition, where the end product of a pathway inhibits its own synthesis. Beyond repression at the operator, the TRP operon employs a second regulatory mechanism called attenuation. This involves the leader sequence, known as TRPL, located upstream of the structural genes. The leader contains a 28 nucleotide attenuator sequence that can fold into alternative stem loop structures. One structure forms a terminator, halting transcription. The other forms an anti-terminator, allowing transcription to continue. This mechanism enables the cell to finely tune tryptophan production based on intracellular availability. The leader region includes 4 complementary sequences that can pair to form 2 mutually exclusive hairpins. If segments 3 and 4 pair, a terminator hairpin forms, halting transcription. If segments 2 and 3 pair instead, an anti-terminator hairpin forms, allowing transcription to proceed through the structural genes. This pairing depends on ribosome position and tryptophan availability, essentially linking transcription and translation in real time. This slide introduces the attenuation model, a sophisticated form of gene regulation in bacteria. As RNA polymerase begins transcribing the TRP operon, ribosomes simultaneously start translating the leader peptide, which includes 2 tryptophan codons. If tryptophan is plentiful, the ribosome quickly translates these codons and reaches the stop site, freeing the RNA to fold into the terminator hairpin. This causes RNA polymerase to fall off the DNA and prevents the expression of the TRP structural genes. When tryptophan levels are high, the ribosome does not stall at the tryptophan codons in the leader sequence. Instead, it smoothly translates the leader peptide and reaches the stop codon located between segments 1 and 2. After termination, the ribosome dissociates, allowing segment 1 to pair with segment 2, and more importantly, segment 3 to pair with segment 4, forming the 3 to 4 terminator hairpin. This structure causes RNA polymerase to pause and terminate transcription, preventing the synthesis of unnecessary tryptophan biosynthesis enzymes. In contrast, when tryptophan is scarce, the ribosome stalls at the 2 TRP codons in segment 1 of the leader peptide. Because the ribosome blocks segment 1, it cannot pair with segment 2. This frees up segments 2 and 3 to pair, forming an anti-terminator hairpin instead of the 3 to 4 terminator. As a result, RNA polymerase continues transcription of the structural genes, enabling the cell to produce the enzymes needed for tryptophan synthesis. A regulon is a group of 2 or more operons that are regulated by a common regulatory protein and its associated effectors. These operons may be dispersed throughout the genome but are coordinated in their response. Regulons allow bacteria to efficiently control multiple genes involved in the same pathway function or environmental response, such as nutrient acquisition, stress adaptation, or sporulation. This slide provides a helpful video resource that explains the attenuation mechanism in detail. The video begins with foundational concepts and then demonstrates how transcription and translation are coupled in bacteria. For best understanding, it's recommended to start watching around the 12-minute and 26-second mark. where the concept of attenuation is thoroughly broken down. The final slide recaps the dual-level regulation of the tryptophan operon. At the coarse level, the TRP repressor binds the operator to block transcription when tryptophan is abundant. At the fine level, the attenuation mechanism uses RNA secondary structures in the leader sequence to fine-tune transcription based on real-time tryptophan levels. Together, these mechanisms ensure that tryptophan biosynthesis enzymes are only produced when truly needed, conserving energy and resources.
Hi, it's Mr. Anderson, and welcome to Biology Essentials Video 31. This is on gene regulation. In other words, how we express a gene or not, how we make a protein or not. And I wanted to start with an organism that lives in our intestine called E. coli. E. coli is interesting in that it eats whatever we eat. And so if I eat bacon for breakfast, it has to break down the proteins and the lipids in the bacon. If I have cereal, it has to break down the carbohydrates. If I have milk, it has to break down the lactose. And so what's interesting about an E. coli is that it can go from an organism that has 0 proteins to break down lactose to one where 50% of the weight of E. coli is simply enzymes that deal with lactose. And it can do it like that. And so how do they do that? They do that through gene regulation. And so in this podcast, I'm going to talk about gene regulation. Before I do that, I want to get some terminology that I'm going to use a lot out of the way. And so a regulatory gene is going to be a gene on the DNA that regulates another gene somewhere farther down. A regulatory sequence will usually be found just above the gene. And so what do I mean by that? So if DNA looks like this, the gene generally will be down here that we want to express or not. A regulatory gene will be somewhere else in the DNA. It secretes something called a regulatory protein, which then can grab onto a regulatory sequence. An example of a regulatory sequence I'll talk about is called a promoter. Once this is all fit up, then we can have an RNA polymerase actually make the gene. And so I'll go through that terminology, but when I'm talking about regulatory gene, regulatory sequence, those both deal with DNA. But when I'm talking about a regulatory protein, that's coming from somewhere else to help express the gene or not. As far as gene regulation examples, most of what we know now comes from bacteria. And so I'll talk about positive and negative control. Positive control example I'll give you is the lac operon. That deals with lactose. And the negative control is the trip operon, deals with tryptophan. And then finally, I'll show you what we know about eukaryotic gene regulation and how they use transcription factors as activators, repressors to either express a gene or not. And so this is how genes are made or expressed, remember. We start with DNA, and that DNA eventually makes messenger RNA, which eventually makes proteins, and those eventually make you. And so any step along the way, we can actually regulate the gene. So we can regulate it post-translationally, post-transcriptionally, so we can do it everywhere. But in general, most of the regulation I'm talking about is just going to be from DNA to messenger RNA. Do we express the gene or do we not? And so this is generally what goes on. We've got a gene like this. Upstream of that, we have a regulatory sequence. An example of this in eukaryotes would be the TATA box. And the reason it's called a TATA box is you have a thymine, adenine, thymine, adenine. And on the other side, you'd have TATA backwards, complementary to that. And so this is simply a sequence above the gene that allows the RNA polymerase to get on. And so we have a regulatory protein coming from somewhere else, remember, another regulatory gene, maybe downstream or upstream from this whole gene. The regulatory protein, an example, could be the TATA binding protein. This is found in us. It'll grab onto the TATA box. Let me get rid of the writing. And it allows RNA polymerase to grab on and actually express that gene. And so if we didn't have the sequence, if we didn't have the regulatory protein, we couldn't make the RNA polymerase and we couldn't make the protein. And so that's basically how genes are regulated or not. And so let's talk about how this works in the lac operon in bacteria, because this is the first one that we really started to understand. So the way they tweak what I just said is that the neat thing about bacteria is instead of just having one gene, they'll have a number of genes. And so they'll have 3 genes. All the genes required to deal with lactose will be put right next to each other. And so we've named those the LAC-Z, Y, and A gene, but they each make a protein and they each help break down lactose. Above that, they'll have a regulatory sequence called a promoter. Remember, that's going to be where RNA polymerase grabs on. And the other thing they'll have in an operon is called an operator. An operator sits right between the promoter and the genes, and the way I like to think about it is it's like an on-off switch. And so it can either be set in an on position or it can be set in an off position. And so it regulates whether or not we turn the genes on or we don't. The other thing that I'm going to add here is something called a repressor. A repressor will plug right into the operator, so it's going to fit right here. And as long as the repressor is available, RNA polymerase can't get on. And so this would be when the pressor is here, the operator is now in the off position. In other words, we can't make these genes. This repressor I'm going to show you is showing what's called positive control. And what do we mean by positive control? What I said was when lactose shows up, we want to make all the proteins to break down and deal with lactose. And right now there's no lactose present, and so it's off. But let's say lactose shows up. In other words, I drink a glass of milk. Now there's lactose. So the lactose shows up. The lactose, you'll notice, is going to fit perfectly into that repressor. And when it fits in the repressor, it changes the conformation or the shape of that protein. In other words, now the repressor doesn't fit in the operator anymore. In other words, it lacks these little prongs, we'll say, that fit in the operator. Okay, so lactose is present, repressor is now off. Well, who can grab on? RNA polymerase now fits. RNA polymerase can fit. There's no repressor. RNA polymerase is going to run down. It's going to make each of the messenger RNAs for each of those lac genes. Each of those are going to make a protein, and each of those are going to break down the lactose. And so we can deal with the lactose and we can metabolize that lactose. So now the lactose is gone, what's happening to our repressor? It's going back to that original shape. And so it's a cool way we can have positive control. If lactose shows up, then we make all the proteins that can actually deal with that lactose. Shows up again, we're going to get rid of that repressor, and we continue. And so that would be positive control. What's an example of negative control? Well, a negative control we learned about from the trip operon. Trip operon, instead of having just 3, it actually has 5 different genes, but they're put right next to each other in the same way. The way it works is it's actually off when tryptophan or whenever that chemical is present. And so tryptophan, remember, is an amino acid. We need it to make proteins. And so bacteria, as long as tryptophan is present, they don't want to have to make tryptophan on their own. And so the way it works here is that the actual tryptophan fits in the repressor. It gives it a shape that actually blocks the RNA polymerase, or the making of those proteins. But let's say, for example, that your diet doesn't have tryptophan, so you're not getting tryptophan in your diet. What does the bacteria do? Well, now the repressor is going to change shape. It's going to change shape. That allows RNA polymerase on. RNA polymerase is going to quickly make those 5 proteins, and then those 5 proteins are going to make more tryptophan. So the tryptophan fits again, and it's going to turn off. And so what that gives us is really cool control as far as a bacteria goes on if we have positive control in the case of lactose whenever it shows up, then we want to break it down, or tryptophan control, negative control, when it's there, then we want to turn it off. So simple, neat engineering solution to a real-world problem. Now, we don't have operons. Remember, in between each of our genes, we'll have long stretches where there's actually junk, quote-unquote, DNA. And so the way it works in us is a little bit different. We use what are called transcription factors. So RNA polymerase is here, but RNA polymerase can't get on until we have a number of transcription factors present. And so let's say we want to make a protein as well in eukaryotes. To do that, we have regulatory factors. Those are actually made by regulatory genes. They could be somewhere else in the DNA, or they could even be outside the nucleus in the cytoplasm. So in order for us to transcribe a gene, it takes a little bit more, it's a little more in depth. First of all, the transcription factors will allow the attachment of RNA polymerase. We'll have other transcription factors that will actually hold it in place, but you can see that we're not actually making the gene. And so in order to make the gene, the DNA upstream of that will actually have to fold. It'll get more transcription factors, and you'll notice that still nothing's going on. We still don't have transcription until that actually folds back and activates that RNA polymerase. Can go now and make that protein or make the RNA and then eventually make the protein. And so how does it work in us? We don't really have these on-off switches. I don't know if you can see my hands. But what happens is the DNA actually folds back on itself, and it can activate genes in other places along the genome. And so it's a different form of control. It's a more complex form of control, but it really requires input from all these other transcription factors. So that's gene regulation, and I hope that's helpful.